Genome interpretation is the umbrella term comprising all the scientific efforts that try to model the relationship between genotype and phenotype. And indeed, being able to precisely understand how our genotype lead to the observed phenotype would be crucial for example, molecular biology, genetics, and could open a new era for precision medicine and agricultural technology. Existing genome interpretation models are mostly based on statistical approaches, such as polygenic rescores in humans or the Bloop family of models for cattle and crop breeding. Those methods were developed before the next generation sequencing explosion, and therefore they mostly rely on SNP array data, such as the one used for genome-wide association studies. The main issue with SNP array data is that they do not sample every locus in our genome, and they take instead a quite sparse picture that could look like this. But if we factor in the true degree of sparsity, it's more likely to look like this. And as you can see, it's quite hard to figure out what's going on from such a sparse picture. Indeed, the concept of SNP array is that it's very unlikely that the causative variants for any phenotype are in there, but the observed variants will be in linkage disequilibrium with the truly interesting one. On the other end, whole genome and exome sequencing are theoretically able to sample every single variance with respect to a reference genome, and so they are potentially much more informative. Now that thanks to next generation sequencing, we start to have a truly huge collection of data, we unfortunately discovered that the bottleneck mostly shifted from a data availability issue to a data interpretation issue. Because even though the relevant variants are in there, they are still hidden in the plethora of neutral variants that drive regular phenotypic expression, leading us to the proverbial needle in the haystack problem. Moreover, genomic data are partially structured data that can be extremely huge, and so they are not trivial to use as input in machine learning or bioinformatics pipeline. Our answer to those challenges are fundamentally based on three tenets. The first is that the big data size and the peculiarities of genomic data should require the development of tailor-made neural network models. Those models should be able to perform data-driven inference in which, for example, the inheritance pattern should emerge from the data. And finally, our assumption is that the genotype to phenotype relationship can be arbitrarily complex, arbitrarily nonlinear for all the non-trivial traits. In this study, we focused on Arabidopsis thaliana, which is a well-studied small flowering plant. It has a fairly small genome, around 125 megabases, divided into 5 chromosomes, but it has a number of genes that is comparable with humans. We collected 1,000 publicly available whole genome sequencing Arabidopsis samples from 46 nations, and we annotated them with 288 phenotypes from the phenotype. Those phenotype databases. Those are real-valued databases. The real-valued phenotypes they describe quantitative traits, for example, morphological developmental traits such as the seed yield or the leaf number or the concentration of metabolites in various tissues. And our goal was to predict those phenotypes from the genome, and the first challenge we had to face was to find a way to encode the sparse genomic information in the VCF files in some sort of machine learning understandable format. To do so, we parsed the VCF with ANOVAR, categorizing each variant into one of 17 functional classes, and then we tried to summarize the mutational damage carried by each gene by counting how many of each type of variant is mapped on each gene. And then we represented each sample as the collection of all those histograms categorizing the gene mutational damage. On top of this gene-centric representation of each sample, we built a neural network model. And the first step is, for example, to use a shared G neuron that reads the description of each gene and altogether gives a minimal latent representation describing each Arabidopsis sample. This latent representation is then used as input for the final P layer that concurrently predict all the phenotypes. Among 288 phenotypes, we were able to predict 75 of them with a Pearson correlation greater than 0.4 and a Bonferroni corrected significant p-value. To give an idea of how they may look like, here I show 4 of them in clockwise order. You have leaf number, root length, the fruit number, and the days to flowering time. We also wanted to check that our model is not just able to predict some sort of average phenotype for the samples belonging to each country. And so, because our goal is to build a model that is able to predict even subtle dynamics, phenotype dynamics within Arabidopsis subpopulations. And so we stratified our prediction by country. Here I show a couple of phenotypes for example, Swedish, Italian, Spanish, and Russian samples. We can see that correlation may be slightly lower, but they are still clearly present. Moreover, we also wanted to verify that our genome-wide multitask approach is actually beneficial with respect to more conventional models that, for example, consider one phenotype at a time and they predict that. So we compared our prediction with several neural network models that we developed. Each of them predict one phenotype, and they take as input only the genes that are already known to be associated to each of those phenotypes as extracted from the ARA-GWAS database. And we show in the paper that in 77% of the cases, actually, our multitask prediction was truly beneficial. Finally, I must say that thaliana belongs to a family of, let's say, machine learning models in which, in order to solve increasingly complex tasks, we build increasingly complex models. And incidentally, this leads to models that are more and more difficult to interpret. They become quite obscure. And so to work towards explainable AI, we use the smooth-grad flavor of the saliency maps methods to interpret machine learning models. And in short, saliency map compute the gradient of the predictions with respect to the input. And they are mostly used, as you can see on the left, in computer vision, for example, to highlight the most salient, the most relevant pixels when it comes to, for example, object detection. In the context of genome interpretation, we can use the same approach to, for example, highlight the most relevant genes when it comes to the prediction of a certain phenotype. We took a lot of steps in order to ensure consistency in those interpretation results, and then we performed a good term enrichment analysis, and in the end, we were able to identify 36 genes associated with flowering time, 6 of which were already known in literature. And just to give an idea of how this analysis may look like, here I'm showing the 50 more activated genes when it comes to predict flowering time with respect to the background activation level. That is the last box plot on the right. And that's it. So a big thanks to my collaborators in Leuven, in Torino, and in Versailles. And thank you for your time as well.
So we have time for questions.
Hi, it was very interesting indeed. I was wondering, when you set up this training protocol, you have to make sure that the training and test data set are not overlapping. So in terms of, then you need to make sure that the phenotypes that you have in the test set are different from the ones that you have in the training set. And how do you measure the distance similarity of phenotypes? Because they could be correlated, they could be very similar.
Very interesting question. Thanks. I mean, we consider that, so the phenotypes that we are predicting are our labels. And so, I mean, they were present. So, for example, flowering time, it was in the training and in the test, because, I mean, it's our label. What we try to figure out is, for example, is there a relationship between how population of Arabidopsis are clustered and how it influences the generalization. And indeed, it's very difficult to generalize between the biology of Swedish Arabidopsis and the Spanish ones, because as our plant biology says, plants don't run, and so their genome run in the genome space. So they adapt a lot, and so it's very difficult. But in general, the phenotypes were the same. We trained to predict certain phenotypes.
So, but then the quantity of the phenotype then, is that the target of your prediction? I thought you predict something like this is different leaf morphology phenotype. This is
no, we
root length phenotype or
288 quantitative traits. Like, so
okay,
how the seed yield, the number of fruits, of leaves, and
okay.
so on. The seed dormancy, and yeah. It's a multi-label prediction.